Assalamu alaikum. Dear students, I am here Hafiz Muhammad Ammar, co-teacher for course Microbiology, code BOTN 4128. Today we are here for lecture number 28, entitled Control of Microorganisms. In previous lectures, we were discussing about the methods for the control of microorganisms. In continuation of last lecture, physical methods in microbiological control, we will cover second portion of that method, like radiation, and we will discuss chemical methods for the control of microorganisms. Ionizing radiation has many industrial, military, and medical uses. Its usefulness must be balanced with its hazards. For example, at one time, assistants in shoe shops use X-rays to check the child's shoe size. But this practice was halted when the risks of ionizing radiation were better understood. So, neutron radiation is essential to the working of nuclear reactors and nuclear weapons. The penetrating power of X-rays, gamma, beta, and positron radiation is used for the medical imaging, non-destructive testing, and a variety of industrial gauges. Radioactive tracers are used in medical and industrial applications, as well as biological and radiation chemistry. Alpha radiation is used in static eliminators and smoke detectors. The sterilizing effects of ionizing radiation are useful for cleaning medical instruments, food irradiation, and the sterile insect techniques. It is often used to sterilize pharmaceuticals and disposable medical supplies like syringes, surgical pipe gloves, catheters, sutures, and patty plates. It is also used to retard spoilage in seafoods, meats, poultry, and fruits. In this technique, the medical instruments pass through the radiation chamber, and the exposure to the radiation for certain time that make it sanitize. If we discuss about the procedure, when particularly the frozen products, the product is stored in a refrigerator and freezers. This product electron beam sterilize the packed product as it moves through the chamber. The increase in temperature is minimal in this technique, and sterilization is completed in just a minute. And this is only possible through electron beam sterilization. Then this product is returned to the refrigerator or freezer, and this means the product spent the last amount of the time removed from cold storage. The dose is verified, and the refrigerated product, frozen product, is now released to the shipments. Similarly, other product materials like antibiotics, hormones, and other biological products are being sterilized by using these radiation techniques. Another technique is microwave radiation. In contrast with the gamma, X-ray, and UV radiation, microwave radiation falls in the long wavelength end of the electromagnetic spectrum. It has wavelengths of approximately 1 millimeter to 1 meter, a range that includes television and police radars. Microwave oven frequencies are tuned to match energy levels in water molecules. In the liquid state, Water molecules quickly absorb the microwave energy and then release it to the surrounding materials as heat. Thus, materials that do not contain water, such as plates made up of paper, china, or plastic, remain cool while the moist food on them becomes the heated. For this reason, the home microwave cannot be used to sterilize items such as bandages and glassware. Conduction of energy in metals leads to problems such as sparking, which makes most metallic items suitable for microwave sterilization. Bacterial endospores, which contain almost no water, are not destroyed by microwaves. However, a specialized microwave oven has recently become available that can be used to sterilize media in just 10 minutes. It has 12 pressure vessels, each of which holds 100 ml of medium. Microwave energy increases the pressure of the medium inside the vessels until sterilizing temperatures are reached. Caution should be observed in cooking foods in the home microwave oven. Geometry and differences in the density of the food being cooked can cause certain regions to become hotter than others, sometimes leaving very cold spots. Consequently, to cook foods thoroughly in a microwave oven, it is necessary to rotate the items either mechanically or by hand. For example, pork roasts must be turned frequently and cooked thoroughly to kill any cysts of the pork roundworm. If you fail to kill such cysts, could lead to the disease trichinosis, in which cysts of the worm become embedded in human muscles and other tissues. All experimentally infected pork roasts, when microwaved without rotation, showed live worms remaining in some portion at the end of the standard cooking time. Visible light. Ultraviolet portion of the light spectrum includes all radiations with the wavelength from 100 to 400 nanometers. It has low wavelength and low energy. Its microbicidal activity of UV light depends on the length of the exposure and the duration of the exposure. As greater the exposure, longer the cidal activity, and it also depends on the wavelength of the UV which is used. I hope you have already discussed UV light in previous lecture. And the most microbicidal activity of the wavelengths of UV light lie in the 260 nanometer to 270 nanometer range, where it is absorbed by the nucleic acids. In an experiment conducted as shown in the figure, the E. coli bacteria was used for the study. The impact of the visible light and UV lights was observed. A molecule encoded with PBBIFE03, as shown in the figure here, when exposed to the UV spectrum of the light, it usually it deregulates the gene in E. coli cells that are involved in the cell membrane damage and oxidative stress responses. When it was exposed to the UV light, it was observed cell leakage of nucleic acids or proteins, as shown in the here in the picture. You can see protein damages due to the exposure of a scavenger, you can say scavenger of hydroxyl radical group, which was released due to the UV light impact, and this exposure damages the cell membrane and protein covering the nucleic acids. And it was observed the transcriptomic analysis indicated that a complete inactivation was reached within 30 minute to 90 minute exposure to the UV spectrum. After the consecutive cycles, this molecule photocatalyst exhibited a strong antibacterial efficiency. So we can say the strong visible light has the greater impact on microcidal activity. In terms of the mode of action of UV light, if we discuss, UV light is absorbed by microbial DNA. And what does it do in the microbial DNA? It causes adjacent thiamine bases on the same DNA strand to covalently bond together, forming what are called as thiamine-thiamine dimers. As the DNA replicates, nucleotides do not complementary base pair with the thiamine dimers, and this terminates the replication of the DNA strand. However, most of the damage from UV radiation actually comes from the cell trying to repair the damage to the DNA by process called SOS repair. In very heavily damaged DNA containing large number of thiamine dimers, SOS repair is activated as kind of last ditch effort to repair the DNA. And the same process is reversed when the DNA exposed to the visible light, and the process reversed, DNA repair, the thiamine bond broken down, and DNA completes its normal replication process. What the process, recovery process to the due to the exposure of the light, visible light, is called photoreactivation. An important consideration when using UV light is that it is very poor penetrating power. Only microorganisms on the surface of the material that are exposed directly to the radiation are susceptible to destruction. UV light can also damage the eyes, cause burns, and cause mutation in the cells of the skin. Part 3. Chemical methods. Physical agents are generally used to sterilize objects. Chemicals, on the other hand, are more often employed in disinfection and antisepsis. The proper use of chemical agents is essential to laboratory and hospital safety. Chemicals also employed to prevent microbial growth in food, and certain chemicals are used to treat infectious diseases. Many different chemicals are available for use as disinfectants, and each has its own advantages and disadvantages. In selecting an agent, it is important to keep in mind the characters of the desirable infectant. What it should be effective against a wide variety of infectious agents like gram-positive, gram-negative bacteria, acid-fast bacteria, bacterial endospores, fungi, and viruses, at low concentration and in presence of organic matter. Although chemical must be toxic for infectious agents, it should not be toxic for people or corrosive for common materials. Some chemicals are used despite their low effectiveness because they are relatively non-toxic. The ideal disinfectant should be stable upon storage, odorless or with pleasant odor, soluble in water, and lipids for penetration into microorganisms. It have a low surface tension so that it can enter cracks in the surface and be relatively inexpensive. One potentially serious problem is the overuse of antiseptics. For instance, the antibacterial agent triclosan is found in products such as deodorants, mouthwashes, soaps, cutting boards, and baby toys. Unfortunately, the emergency of triclosan resistant bacteria has become a problem. For example, Pseudomonas aeruginosa actively pumps the antiseptic out of the cell. There is now evidence that extensive use of triclosan also increases the frequency of bacterial resistance to antibiotics. Thus, overuse of antiseptics can have unintended harmful consequences. The potency or effectiveness of a chemical antimicrobial agent is affected by time, temperature, pH, and concentrations. The death rate of organism is affected by the length of the time of the organisms are exposed to the antimicrobial agent. The death rate of organisms subjected to a chemical agent is accelerated by increasing the temperature. Roughly increase in the temperature 10 C doubles the rate of chemical reaction and increases the potency of the chemical agent. In figure shown in the slide, you can see the action of the surfactant. The surfactant molecule has ionized into sodium ions and long hydrocarbon chains, whose zigzag covalently bonded trails are able to enter an insoluble substance such as grease. The other end of these molecules has a carboxyl group with a negatively charged oxygen. These negative charges attract the positively charged sides of water molecules, thereby making the attachment insoluble substances soluble in the water, so the substances can be washed away. These are the chemical antimicrobial agents commonly being used in the for disinfection and antisepsis. Phenolics, alcohols, halogens, heavy metals, quaternary ammonium compounds, aldehydes, sterilizing gases, chemotherapeutic agents. In today's lecture, we will only discuss two of the chemical antimicrobial agents, phenolics and alcohols. Phenol was the first widely used antiseptic and disinfectant. In 1867, Joseph Lister employed it to reduce the risk of infection during surgery. Today, phenol and phenolics, such as cryzols, xylenols, and orthophenylphenols, are used as disinfectants in labs and hospitals. The commercial disinfectant Lysol is made up of mixture of phenolics. These phenols act by denaturing protein and disrupting cell membranes. Some phenolics are tuberculocidal, effective in the presence of organic material, and remain active on surfaces long after application. However, they are disagreeable odor and can cause skin irritation. Hexachlorophene has been one of the most popular antiseptic because once applied it persists in the skin and reduces skin bacteria for long periods. However, it can cause brain damage and now used in hospital nurseries only in response to a staphylococcal outbreak. Amphile, which contains amphyl phenol, destroys the vegetative forms of the bacteria, fungi, and inactivates viruses. It can be used on skin, medical instruments, dishes, and furniture. While used on surfaces, it retains its antimicrobial action for several days. Hydrochlorophene and dichlorophene, which are halogenated phenols, inhibit staphylococci and fungi on the skin and anywhere else. It is a good agent for surgical scrubs. It is excellent skin disinfectant. First, it was used as antiseptic extensively in the 1960s for daily bathing of infants. And hexachlorophene is absorbed through the skin and travels in the blood to the brain. Baby powder containing hexachlorophene killed 40 babies in the France in 1972. And it is now available in the USA only by prescription. Alcohols. These are the most widely used disinfectants and antiseptics. They are bactericidal and fungicidal, but do not kill the spores. Some lipid containing viruses are also been destroyed by using these. The two most popular alcoholic germicides are ethanol and isopropanol, which are being usually used in about 70 to 80% concentrations. They act by denaturing proteins and possibly by dissolving membrane lipids. A 10 to 15 minute soaking is sufficient to disinfect thermometers and small instruments. When it is mixed with the water, alcohols denature proteins. They are also lipid solvents and dissolve membranes. Ethyl and isopropyl alcohols can be used as skin antiseptics, while isopropyl alcohol is more often used because of legal regulation of ethyl alcohol. It disinfects skin where injections will be made or blood drawn. Alcohol disinfectants does not sterilize skin because it evaporates quickly and stays in contact with the microbes for only a few seconds. It also does not penetrate deeply enough into the pores in the skin. It kills vegetative microorganisms on the skin surface but does not kill endospores, resistant cells, or cells deep in the skin pores. If we are looking into the properties of the chemical antimicrobial agents, the hidden parts will be discussed in later lectures. In this lecture, we are discussing about alcohol and phenol. You can see alcohol denature protein when mixed with water, as already discussed. It is being used to disinfect skins, and propylene glycol can be used in the aerosols. Similarly, phenols, which are disrupting membranes, denature proteins, inactivate enzymes, not impaired by organic matter. These are used to disinfect surfaces and destroy discarded cultures. So these are the references where from this lecture was prepared. You can also study these books for further studies. Thank you very much.